Jatropha curcas is known also as Barbados nut or tuba-tuba. It is a shrub belonging to the Euphorbiaceae or spurge family.
These plants are found in tropical countries throughout the world. They are grown as ornamental plants and they are found in gardens and public areas, and therefore will be easily accessible. The plant is particularly attractive to children. Chemistry of poisoning. Quercetin, a phytotoxin toxalbumine found mainly in the seeds and also in the fruit and sap. Phytotoxin or toxalbumins are large complete protein molecules of high toxicity. They are toxic because they inhibit protein synthesis. It is a dimer made up of two subunits, simply designated A and B. A is the actual toxin, while B is a lectin, carbohydrate-binding protein, that aids in the entry of the toxin in the cell membrane. A then breaks down critical proteins and essentially shuts down the cell. Purgative oil. The seed yields 40% oil, which contains small amounts of irritant quercetinolleic acid. Toxicokinetics. Phytotoxins are well absorbed from the gastrointestinal tract and metabolized in the digestive system. Signs and symptoms. Vomiting, abdominal pain, and diarrhea occur within 60 to 90 minutes of a significant ingestion. The vomitus and feces may contain blood. Severe gastroenteritis may result massive fluid and electrolyte loss. Hypotension with a fast, weak pulse and shock may also occur due to fluid and electrolyte loss. Electrocardiographic abnormalities and rapid but shallow breathing is seen. Liver damage may occur in serious cases. There may be increased ALT, total bilirubin, and AST. CNS depression either as a direct result of toxin or secondary to hypotension. Mydriasis, dry mouth, flush hot dry skin, and tachycardia are some of CNS symptoms. Tox-albumin poisoning may also produce fever. Management. Diagnosis. It is based on the history of exposure and is suspected when plant material is seen in vomitus. Identification of the plant is essential to proper treatment. Administration of activated charcoal, if available, or ipecac-induced vomiting may be useful for initial treatment. Serum toxin levels are not available for most plant toxin. Other useful lab studies include CBC, electrolytes, glucose, BUN, creatinine, and urinalysis. If liver toxicity is suspected, also obtain liver transaminase and prothrombin time. Treatment. Decontamination is indicated for all known or suspected poisoning. There is no antidote. Rehydration, either voluntary water ingestion or IV administration to counteract fluid loss due to vomiting and diarrhea, is critical. Treatment is essentially symptomatic and supportive. The more critical analysis and investigations are fluid and electrolytes, acid-base status, full blood count, and renal and hepatic function. Monitor level of consciousness. A specific therapy may be indicated for hemorrhagic gastrointestinal damage. After substantial exposure to tox-albumin containing plants, an observation of up to 8 hours is advised.